and taking blood samples, and there's some genetic studies that they did as well. And I still keep in touch with my teammates, and it's really neat to be involved with people that are very passionate about ecology as well. So that was my first summer, and then this past summer in July, I went down to Princeton, and that was another all expenses paid through this time the Howard Hughes Medical Institute. So there I got to practice. I wasn't outdoors this time, I was totally inside. So I got a totally different experience. And I got to practice techniques in biotechnology that are common in research nowadays, and we got to do some really neat things. So it was focused on genetics, and I'll show you guys one really neat thing we did. This is the class of 2010. So there are teachers from all over the country, and one person from Qatar, actually, that I'm standing next to. So one of the things we did was we analyzed our DNA, and we looked at our combination of genes, and one particular area of the genes, the D1S80, which is used to identify people because there's a lot of variation. And what I found out is I was the only one that actually was what we call homozygous for this particular version. So you can see under me, let's see, there I am. So I only have one line there. So both of my parents gave me the same exact combination, but everybody else had two. So I found out I was kind of unique as far as that. And something else I did was really, really amazing. And something I'm passionate about too is art. So we took bacteria and we transformed them, allowed them to express a protein that fluoresces. And we got to play with a palette of 6 different colors. So we practiced some DNA art, they called it. And I decided to make a butterfly. So you can see some of the colors. The yellow, the green, the blue, and a little bit of the purple. So some of my colleagues were very, very adept at this. I kept it kind of simple. But under UV light, you can see pretty well the colors. And this is a technology that's becoming really, really useful. And one of the great things about this program is that they actually allow you to borrow equipment and bring the kids down to Princeton and get them exposed to some of the lab experiences. So I thought it was a wonderful experience. And again, I'm still in touch with the teachers from there, who were a very passionate group as well, and had a wonderful time. So those are my 2 experiences in New Jersey.
I also did leave the home state. I got an email from the New Jersey Biology Teachers Association about this, the Waksman Student Scholar Program. And it sounds really phenomenal, so I filled out the application, and I got word that I got accepted, and I spent the 2 weeks, the end of June, right up through July 4th, with a group of teachers from all over the country. And actually, my lab partner, John Anderson, he actually teaches over in Pakistan. So just a wide variety of people. The program this year, it's been running for a couple of years. Waksman is on the Rutgers campus at Bush, and I spent 2 weeks there. And the program this year is called the 2010 Hygiene Project. The organism that we're studying, and again, this was very, very highly intensive biotechnology. The organism that they're studying this year is a species of duckweed called Wolfia australana. It's the world's smallest fire proponent. And the first thing they told us, why did we pick the duckweed? And there's 3 great reasons. One, it produces starch, accumulates starch, but much larger quantity than corn, and we're using corn as biofuel. The other thing that the duckweed does is bioremediation. It can grow in contaminated waters and actually can sequester the contaminants like lead and arsenic, and even degrade it. And that's important in New Jersey because I believe we're number 1 in Superfund sites. The website is on the top there. If you go and you Google search the Waksman Student School Program, or go to the website and you'll click on the Scholar Program, this is what you're going to see. And I just took a snapshot of that because it gives an overview of the program, and it shows that the program is really two parts. I went in the summer and spent the 2 weeks, but we're also committed to the academic year to involve students in the same type of work that I did, which is extremely exciting. It's going to be a lot of work, but also exciting. And on the left, there is links to use during the project, but there's also an amazing amount of resources for all the science teachers here and for all the students that are involved. So the nice thing about this program is the students are going to be doing real science, working with real scientists, real data, communicating with them, but that's the general goal of the project. The main goal of this project is to have every student analyze a DNA sequence from the Wolfia genome, from a clone in a library. And the exciting thing is because this organism has never been studied, many of these students are going to find a novel protein. And that sequence is going to get published in an international database, SCBI, which stands for the National Center for Biotechnology Information. Minimally, I did all of that in red twice in the 2 weeks I was there. It was a lot of work, long days. Started at 7:30, we ended about 10 o'clock. We went back to the hotel, we had homework, we would get together and work on our minimally, what they want us to do with students is at least do the DNA analysis, which involves, computer work. Hopefully, and I'm going to try to do that. I teach honors biology. I have about 63 students. I'm going to try to get all of them to do the bioinformatics. Now, that's going to happen after I teach, and they get really efficient in DNA and protein structure and function and protein synthesis. Then they're going to be ready to do the bioinformatics. The professor that we worked with wants them to do practice clones before they actually start doing their own DNA. Hopefully, though, hopefully they also can. I want to get some of the students involved in the actual wet lab technology, PCR, polymerase chain reaction, and then purifying DNA with a technique called DNA mini preps, and then isolating so we can do the sequencing of the Wolfia DNA with doing restriction digests. Either way, there's some microbiology involved on the left where students pick clones from the DNA library. I don't know that I'll do that this year, but what are the clones? And we're not like doing sheep, like making dolly or anything. The clones, very simply, are we start with rod-shaped bacteria, Escherichia coli, very common flora in our intestines, should not be in our hamburger meat. And they have a circular DNA called a plasmid, and we basically make recombinant DNA, and all that is DNA from 2 sources. So you see the black is the DNA from the Escherichia coli, and then this would be the DNA, in our case, from Wolfia. I mean, you can make recombinant DNA with human genes. That's how we genetically engineer human insulin, growth hormone, and a lot of other things. So that's what we mean by a clone. It's really a recombinant DNA. So this, and then you did similar work at Princeton. This is what's going to show up. Is you're going to isolate that gene from the Wolfia, and you run this thing called a PCR, and you get these bands, and that represents the DNA. And on the left is marker. On the left is marker, and that helps us determine the size of our sequence. And if the sequence is over 200 nucleotide bases, we're going to send it out for sequencing. And then we get that back. This is the bioinformatics. We use this software called DSAP, it stands for DNA Sequencing Analysis Program. It takes it's very cumbersome in terms of time, but the way they have set this up, a step-by-step process, I know my students are going to be capable of doing it, although it will take some time. This is one of my clones. Now, the other thing is they're going to learn about science and how important it is to keep good records. And we're going to keep our records and communicate with the scientists at Rutgers by using Google Docs. And you can see this is our school. We're number 57. There's a total of 62 schools, including Rutgers University, involved in the program over the last few years. We're one of just over 20 New Jersey high schools. And we've been assigned a number. We're West Morris Central's number 57, and all the clones that my students, these are the 8 that I did this summer, as we do our sequencing, and we submit this, and if it's novel, they'll send it in to get published.